And thanks for the invitation to give a talk here. So as Anna said, I'm a theoretician, and my charge really is to talk a bit about theory and how aspects of things like epigenetic inheritance might play a role in how we model and understand the evolutionary process. And unlike the other talks you've seen so far today, I'm going to be very light on mechanism at the molecular level. And part of the reason is I want to make an argument that at least at some level we can model and understand at a phenotypic level the consequences, or some of the consequences of epigenetic inheritance without worrying too much about the specific details. But I'll show you what I mean by that as we get there. So I'm going to talk about drug resistance. Rather than talking about theory in sort of an abstract way, I want to present some ideas of how we can model the consequences of epigenetic inheritance within the context of a specific example. And the example is non-genetic or phenotypic, or sometimes called tolerant drug resistance in bacteria. And so I'll give you a bit of history. Everybody probably knows Alexander Fleming. He was a Scottish physician. He was awarded a Nobel Prize in 1945 in part for his role, his part in the role of discovering penicillin as the particular antibiotic. You probably don't know, or most of you probably don't know this guy. He was a contemporary of Fleming, Joseph Bigger. He was an Irish physician, and the year before Fleming got his Nobel Prize, he published this paper in The Lancet, which has come to be kind of the foundational paper in non-genetic drug resistance in bacteria, or what are sometimes called persister cells. And what he did in this paper, basically, is he showed that even in the absence of genetically determined resistance in bacteria, when you apply at least penicillin in this case, you eradicate very quickly a lot of the bacteria, but there seem to always be a few cells that are left that you can never seem to get rid of, and he referred to these as persister cells. And he argued that they were a phenotypic form of resistance rather than a genetically determined form of resistance. And I'll show you some experiments. So there's been a huge amount of work that's been done in this area, and I'll show you some experiments that have been done since then, because it more clearly illustrates this idea that he was getting at. So basically the idea here is you have a population of bacteria, you apply an antibiotic to it, and this is measured on a log scale here. So this is a log scale on the vertical axis. If there was a constant killing rate of bacteria, you'd see a linear decline in the population size of the bacteria. But instead you see this saturation, where you get some bacterial cells here that tend to remain after you're finished applying this antibiotic. And these are what Bigger referred to as persister cells. And what he reasoned was, well, you could imagine there was a population of cells to begin with, and there was genetic variation, some genetic strains there that were resistant. If that was true, then these cells are presumably the genetically resistant ones. And so if you took that population and reconstituted a large population size and applied the antibiotic again, you should end up with a pattern like this, where you don't see any decline once you apply the antibiotic. And what he found instead was, if you do this repeatedly, day after day, you regenerate the very same sort of pattern, where I take the persister cells that are left at the end, and then I apply the antibiotic again, and you see exactly the same sensitivity, but there's always some that remain. And so he argued that because of the time scale of this must be some kind of phenotypic resistance rather than a genetically determined resistance. And there's been a ton of work, as I said, on this since. I was going to show you a video here, but it didn't come through on the PDF format of the talk. But we now know that he was right in terms of these being phenotypically resistant cells. And in fact, the persister cells in many cases are dormant cells, cells that no longer replicate. And in this video, what I was going to show is this cell here in the middle is a persister cell. So people have done these single-celled studies. And the video would show when the antibiotic is applied, all of the cells around it that are replicating get killed off very effectively, but that dormant cell is unaffected. And then when the antibiotic is removed from the system, that dormant cell can reawaken somehow, and it begins to replicate. And so this persister cell phenomenon seems to involve this aspect of dormancy. So what I'm going to do in my talk is I'm going to use this as an example, or a potential example, of what I'll call epigenetic inheritance, but really I'm going to use that term in a very loose way. And I'm going to first present four main kind of qualitative empirical findings of persister cells, what people generally have seen in terms of patterns of persister cell formation. And then I'll tell you a bit about what the main evolutionary hypothesis is that's been put forward to explain these patterns. And the main thing I want to do is this last part here, which is argue that maybe there's a simpler way to explain these results, and that has to do with evolution under non-genetic, or what I'll call epigenetic inheritance. So the first observation, the first empirical pattern is what I already showed you. It's this pattern of basically phenotypic dormancy, where there's always a small fraction of the population that seems to be dormant and is able to resist antibiotics. The second pattern shows that the rate at which persister cells are formed seems to depend on the population dynamics. So I'll show you a couple of different plots like this. This top line here is the population size of a bacterial culture over time. And at each point in time, they took a sample from the culture, and then they basically did that experiment that Bigger did. You apply an antibiotic, and you measure how many persister cells are left. And so by doing that, you can get this bottom curve, which is the number of persister cells that are found in that culture over time. And the idea behind this plot here is to show that, well, you see a very low level of persister cells, and then all of a sudden something happens and the number of persister cells increases, and it seems to be associated with an approach to stationary phase in these bacterial cultures. And so the argument is maybe there's a phenotypically plastic response here. As resources get depleted, the bacteria are now turning on this persistence phenomenon and they're becoming dormant. And so this has been referred to as a type 1 form of persistence. And that contrasts with what's been called type 2, where if you do the experiment with under different conditions, this is again the total population size, but now you can see that the population always contains a constant fraction of persister cells. And so the argument here is that they're not responding to the environment in any way. They're just producing every time there's a replication event, there's a certain probability that a dormant or persistent cell will be formed. And that's been referred to as a type 2 type of persistence. So that's the second observation, that the formation of these things seems to depend on the population dynamics. And sort of related to that actually, the third observation is that if you maintain a bacterial culture in exponential growth phase, so you take a culture like this over here, and before it reaches stationary phase, you transfer it, and you keep doing that, so the cells are always in exponential growth phase, you can eradicate the persisters from the population. And that's what the data on the right-hand side shows here. So these dark gray bars are the total population size. The 3 bars are 3 different replicates. The light gray bars are the population size of persister cells, and this is the serial dilution that happens as you go along. And this horizontal line is the detection threshold. And so you can see after a few serial dilutions, you eventually eliminate all of the persister cells, or at least they go below a detectable threshold, provided you maintain the culture in this exponential growth phase. And so again, there's a suggestion, maybe this is because these are what we would call type 1 persisters. The environment is good, there's lots of resources, so persister cells aren't even formed in this kind of situation. So that's the third observation. And the fourth observation is that there is actually some, and quite good, direct evidence in a variety of different experiments that show that persister cell formation can be triggered by certain environmental stimuli. And this is just one experiment that shows that. So here, again, is a measure of, for a wild type, I think it was E. coli in this case, a wild type, the number of persister cells that's the fraction of persister cells that are formed when you apply an antibiotic, ampicillin in this case. And then if you do the same experiment, but you first stress the bacteria by applying an acid, and then you do this assay to measure the number of persister cells, you see there's an increase in the fraction of persister cells that are formed. And so again, an argument that this is maybe a type 1 sort of persister cell formation, where they're responding to some environmental cue and going dormant when the conditions are bad. So those are the 4 observations. That persister cells are a dormant transitory phenotypic state, that the rate of formation seems to depend on the population dynamics, and that you can eradicate them if you maintain them in a bacterial population in a state of exponential growth, and that stress, at least certain stresses, and for certain bacteria, sometimes seems to increase persister cell formation. And now the main evolutionary explanation for this, it's actually a bit muddied in the literature, but it involves a combination of phenotypic plasticity and bet-hedging. And so, roughly speaking, the argument is that at least for type 1 persisters, what's happening is there's some kind of environmental trigger that represents a stress in the environment, and when that stress is experienced, that's when cells generate persister cells, and so they can remain dormant and withstand this stress. And so it's a form of adaptive phenotypic plasticity that's genetically encoded in the bacteria. In the case of type 2 persisters, the argument is that it's a bet-hedging strategy. So it's no longer a response to any sort of environmental stimulus, but every time there's a replication event, a certain fraction of the bacteria will become persister cells. And the idea is that that's a sort of bet-hedging strategy. If the environment is fluctuating and it happens to be bad in a particular year, always having this small number of bacteria in this dormant state is a safe sort of bet to play in order to ensure that you survive these bad periods. So given those two hypotheses, it's not surprising then that people are interested in trying to find the genes that code for plasticity in this case, or the bet-hedging strategy. And so there's been a lot of effort devoted to looking for the genes for persister cell formation through basically screening of knockout libraries. And one of the interesting things is that when people have done that, there doesn't seem to be a gene for persister cell formation. No matter what gene is knocked out, persister cells are always formed. It affects the fraction of cells that become persisters, but there are no knockouts where they can remove persister cell formation. And so you might initially think that that kind of argues against this being a genetically determined plasticity or bet-hedging strategy, but interestingly it's been taken as an argument that that must be what it is, because now the argument is that this is so important evolutionarily that there are multiple genetically redundant pathways that code for this plasticity or this bet-hedging strategy. And what I want to argue is that maybe there's a different explanation that involves, again, what I'll call epigenetics, but I'm going to use that in a very loose term, or in a loose sense. And I'm thinking of anything, so these are bacteria, and they're typically replicating by binary cell division, and so I'm thinking of everything outside of the genome as being part of what I'm going to call the epigenome in this case. Now, typically, evolutionarily, how we think of populations is, so here are two parents, and they differ genetically in terms of the color that I've given the chromosomes here. And we can model evolutionary change. There's a wealth of theory in the literature in population genetics that allows us to understand how we expect populations to evolve. And one way to characterize that is through what's called Price's equation. And I won't go into the details of it too much, but in the context of genetic evolution, there are basically two important pieces. There's a piece that tells you how the different genes are related to fitness, and then there's a piece that's associated with the fidelity of transmission of those genes. And maybe there's mutation that interrupts that fidelity or recombination. This piece is typically relatively small if we're thinking of allele frequency changes, and because genes are very stable, and so most of the stuff people are interested in is this fitness-related variation. Of course, in bacteria, and we can think of this more broadly in other organisms, there are a bunch of things that get transmitted from parent to offspring. And that's what I'm going to refer to here as the epigenome. So even in the case where there are genetically identical individuals, there are other things that get transmitted to the offspring. And those other things can play a role, and we can model those things, the evolution of those things, in the same sort of way using Price's equation. And so again, you get a component in the equation that's due to differential fitness between different epialleles, if that's what we're talking about, or other aspects of the epigenome. There's a component that's due to differential fidelity of transmission. And now this no longer need be mediated through the sort of typical Mendelian patterns of inheritance. It can be a variety of different things depending on what the substance that's transmitted is. And then there's a component that involves induction from the environment. So the environment can change the state of this stuff, this epigenetic stuff, or whatever it is we're talking about, and that can then potentially be passed on across generations. And so there are these three terms in the case of this non-genetic stuff. And of course, these two equations interact with one another because there can be an interaction between the genetic and the non-genetic stuff. Now, what I want to do is, I'm going to focus only as an attempt to provide an explanation for these patterns in the experimental results for persister cells. I'm going to forget about anything to do with the genetic component, at least for the time being. And I'm going to imagine that we're only going to talk about the non-genetic stuff that gets transmitted from parent to offspring. And so I'm going to construct a model for how the population of bacteria will evolve, and it's going to involve these three components: a piece due to selection, a piece due to differential fidelity of transmission, and a piece due to induction by the environment. Now, in the case of persister cells, you could ask, what is the thing you're measuring? Something's getting transmitted in addition to the genome that presumably is playing a role in whether a cell is a persister cell or not. And as I said, I'm going to be pretty a-mechanistic. I'm not going to worry too much about the details, but I want to, at some level, have a model that at least captures qualitatively what we think must be going on. So people have looked for mechanisms that determine persister cell formation. And one of the things they found is there are certain molecules, in this case a molecule called HIP-A molecule, that when you exceed a certain threshold concentration of that molecule, cells tend to become persister cells. And so what I'm going to do is construct a model where there is something I'm just going to refer to as expression level, or you can think of it as propensity to become a persister cell. And that gets transmitted from parent to offspring through some cytoplasmic element, whatever it is. Maybe it's this molecule itself, or maybe it's a suite of molecules that gets transmitted from parent to offspring. But it's going to be some quantitative measure of the likelihood of becoming a persister cell. And that can be transmitted from parent to offspring. And so we can think of a population of bacteria as having a distribution of different expression levels, and the area under this curve below the threshold is all of those cells that are replicating, they're not persister cells, and the area above the threshold is all of those cells that are dormant, that are persister cells. And so here's a bigger picture of what it is that I'm going to model. So I'm going to keep track of the population density of bacterial cells with different expression levels. And that expression level, whatever it is, can be transmitted with some fidelity from parent to offspring. So as I just said, these are the non-persister cells, and this shaded gray area is the concentration or the expression level above X star at which a bacterial cell will become a persister cell. Now, I'm not going to go into too much of the mathematical details here, but I'll go into a few of the details just to give you a sense of how this sort of model is constructed. There are basically three main dynamical components that change the density of bacterial cells with different expression levels. The first is reproduction. So bacteria with different expression levels can replicate at different rates, potentially. Of course, anything above this expression level X star is not going to be replicating because it's a dormant persister cell. And so that's going to increase the density of bacteria with these different expression levels, except the ones above X star won't change. So that's that first component of the equation, differential reproductive success. There can be transmission of this expression level from parent to offspring, and that can happen with some level of fidelity. So when a parent produces or divides into two offspring, the expression level of those offspring might be the same as the parent, or they might differ from the parent, depending on how you want to model things. And there can also be noise within the parental generation in these cellular processes that alters expression level. And so when parents reproduce, they're going to produce offspring that maybe are different from themselves, and that's going to tend to basically even out this density curve. It introduces noise, basically, and tends to level this curve out. And then the last thing that is incorporated is a kind of environmental induction, which I'll refer to as homeostasis. So within the lifespan of an individual cell, there are biochemical processes that tend to drive this expression level to some homeostatic set point, which I'm labeling with a gamma here. And that's a set point at which cell replication can actually happen. Okay, so those are those three components in that equation that I showed you. Well, here are some of the gory details. So basically, just to show you that there's nothing magical going on, I keep track of the number of bacteria with different levels of expression, and that just comes from those that survive. And then there are births, and births that don't undergo mutation. So mu is the mutation rate here. So some cells, when they give rise to new cells, give exactly the same expression level that they had. Of course, you can generate expression level X from reproduction from all the different cells in the population, depending on how they mutate to that expression level. And then there's this environmental induction piece and some noise. This induction piece that drives the expression level within an individual's lifespan towards this set point. And you can work with an assumption of a small mutation and come up with an equation that gives you a model for how that population density changes over time. And as I showed you in the previous sort of general theory slide, there are the three main terms: this differential reproductive success term, this differential fidelity of transmission term, and then there's this within generation induction term, if you like. This fourth one here just is a term that is random noise and expression level. Now, because all these experiments are involving bacteria in culture, and they involve basically, a lot of them at least, involve reaching stationary phase, you want to couple this to an explicit dynamic of resources. So bacteria are consuming a resource, and as that resource gets depleted, then the bacterial population reaches stationary phase. So this model is coupled to a simple equation for the dynamics of the resource. And what I'm going to do is just go through some predictions that this model makes to show that it can explain those sorts of patterns that we see in those experimental systems that I showed you at the beginning. So first, just a couple of assumptions to highlight how the structure of the model works. The simplest assumption to make is that there's a constant birth rate and a constant death rate, regardless of what the expression level is, provided it's below this threshold, and above that the cells are dormant. So that's how reproduction works in this model. There's unbiased mutation in this model, so every individual, regardless of its expression level, when it replicates, it produces offspring that have expression levels perhaps different from itself in an unbiased way. And then there's this homeostatic process that's always driving the population towards this set point gamma. So I'll just show you what happens. I'll give you 4 predictions that basically roughly mirror those 4 observations that I showed you, and explain them in terms of those 3 dynamical processes that go into this model. So the first thing you can do is run this model and ask what happens. What does the density of cells with different expression levels do over time? And it eventually reaches some equilibrium here where t equals 200, and it's got a peak at this homeostatic set point, but there's some variation in the population. When you apply an antibiotic, then of course what happens is you kill off all the bacteria that are replicating. So you kill off everything that's below this threshold, and all you're left with is this little bit of the population that was dormant when you applied the antibiotic. So that gives rise to this kind of pattern that I showed you in the data. You kill everything off except for these little persister cell population that remains. And then what you can do is take this population, reconstitute it, and then apply the antibiotic again and see what happens. And not surprisingly, when I take that population and I grow it back up again to a high enough population to apply the antibiotic, well, some of these dormant cells eventually awaken. They come out of their persister dormant cell state. And eventually you reconstitute a population that was identical to what it was before. So that when I apply the antibiotic, I get the very same kill curve that I had before. So you end up with a pattern where you get the same chunk of the population left, that is these phenotypic persister cells. And it happens basically because there's a very rapid evolutionary adaptation of the population between this phase and this phase that occurs as a result of this inherited epigenetic stuff, whatever that might be. So you can explain this sort of pattern quite readily using this model because of the speed at which these non-genetic elements can mutate and adapt. A second prediction is you can generate those patterns that show that the rate of persister cell formation depends on the population dynamic. So for example, if there's a very low amount of mutation or a very low amount of noise, so that when cells reproduce, they typically produce the same level of expression as they had, then what you see is the population rapidly grows and reaches near an equilibrium at t equals 500 here. But because there's very little mutation and these cells aren't growing, there isn't much that happens to change the density of those cells initially. So the curve goes up very quickly below the threshold, but there's this sort of lag. Any of the cells that are above this threshold just sit there. And the only way the density of those cells can increase is from other cells that are replicating, mutating, and producing those cells. So if you look at what happens to the fraction of the population that are persister cells, well, it's basically very low initially, and then it starts to increase after the population reaches carrying capacity. Again, simply because this adaptation through a combination of mutation and transmission with differential fidelity of this expression level. And that's in the case where there's a low amount of mutation and a low amount of noise. You can imagine if there was a high degree of mutation or a high amount of noise, then you're kind of homogenizing everything very quickly before the size of the population changes much. And so you basically generate a curve that looks like this, and then the curve with that very same shape just slowly moves upward as the population reaches an equilibrium population size, and that's this top line here. Essentially, you get a constant fraction of the population made up of persister cells throughout the entire growth cycle of the population. And again, it's just due to a combination of adaptation and rapid mutation and noise in this epigenetic component of inheritance. And from that, it's not a big stretch to see that you could imagine eradicating the persister cells by maintaining a population in exponential growth in sort of a similar kind of way. So there were these experiments where you had this serial dilution Well, if the mutation rate is relatively low compared to the replication rate of the bacteria, then there's going to be a lot of replication, and these curves are going to go up very quickly when you're below this threshold expression level, but nothing changes the density of those very much. So as I go through this serial dilution process, eventually I'm going to lose most of these persister cells because they're not replicating, and they're not really being formed at all because of the low level of mutation during the replication of the cells that are actually growing. So if you look at the density, so this is the probability density of cells of different expression levels under different replication rates. As the replication rate gets really high, so if the resources are really abundant, it becomes very skewed so that the fraction of the population that are persister cells goes to zero as the replication rate gets big relative to the mutation rate. And finally, this observation that when you apply a stressor to populations, they sometimes seem to produce a higher number of persister cells. Well, if in the model you allow for the possibility that when I apply a stressor to the population, that increases the amount of noise in replication, or it increases the amount of noise even within a cell's lifespan, then that is enough to give rise to that pattern. And the reason is simply that if I'm in a low stress environment, say, and I reach some equilibrium distribution that looks like this, the area under the curve above this threshold is the number of cells that are persister cells in that situation. And then when I increase the stress, that just increases the noise in the population. And so that homogenizes this curve. And you end up with a curve like this that looks like basically a flatter curve, and so you get a higher fraction of cells that are in this dormant persister cell state simply because of the increased noise. And this is related to a hypothesis Bruce Levin has suggested for persister cell formation, where he argued it's not an adaptive phenomenon, but that it's simply something like mutation. And as you increase stress in the environment, you're effectively increasing some kind of mutation. It's not genetic mutation, but some kind of epigenetic mutation that basically causes cells to be dormant because they're broken, essentially. This is an analogous kind of thing, except that all of these different expression levels, in a sense, are within the maybe, quote, normal range of expression level, and changing the level of stress changes the amount of noise and where they fall on that continuum, but it's all, in some sense, a normal expression level. And I guess the other thing to say is that if this is an explanation for these different empirical patterns, then it's not surprising that you don't see any gene for dormancy or persister cell formation, because there wouldn't be a gene for dormancy. So there are all of these genes, and not all of them are shown up here, that affect the frequency with which persister cells are formed, but you never seem to be able to eradicate persister cells. And that's to be expected in this kind of a hypothesis as well. There's always going to be persister cells because there's a sense in which it's an unavoidable consequence of whatever the cellular transmission is from parent to offspring. But when you look at different mutations, you might have different degrees of persister cell formation because mutations will affect the relative importance of these three ingredients that go into the model that determines the frequency of persister cells. So it might alter the replication rate of bacteria, it could alter the degree of noise, or it might alter the way in which they respond to the environment or go through this homeostatic process as well. So that's an argument that looking at a model that doesn't have really any genetics in it might provide an explanation for these patterns that are well documented now for persister cells in bacteria. Of course, we know there are genes for resistance as well. And so the next thing that we want to do is look at the full model, like I showed you at the beginning of my talk, that's coupling this non-genetic material to the genetic material. Typically what you see, so we've played around with models that look like this, that have a genetic component of resistance and this non-genetic component. And typically what you see is a very rapid adaptation of the non-genetic component to conditions first because of the instability of epigenetic material. That provides a lot of variation for selection to act on. So that can adapt very quickly in the initial phases, but it never really adapts as well as genes would because, again, of that mutability of them. And so over the longer term, genes that underpin the resistance can then move in and spread and replace the non-genetic material that initially evolved to code for resistance. So just to summarize then, what I showed you is that there's a model of what I'm calling epigenetic inheritance in a loose sense might provide in some sense, more parsimonious explanation for these observations. More parsimonious in the sense that it's not an argument based on evolutionary adaptation, really, at least not genetic adaptation. It's rapid evolutionary change in the composition of the epigenetic component of the population that gives rise to these different patterns that we see in these experiments. And it also provides an explanation for why when people have looked for genes for dormancy or persister cell formation that we haven't been able to find them. And I'll leave it there with thanks to Bruce Levin for a number of discussions on persister cells and Fritjof Lüscher for discussions about various aspects of the modeling, and I'd be happy to answer questions.